Hello, everyone. Thank you so much for joining me today. It looks like we've got a good crowd here, so that's exciting. My name is Shannon Holmes. I'm a medical physicist here at Standard Imaging, and I'm very excited to talk to you about designing the Exradin A30 chamber so that we can move towards providing NIST-traceable flash therapy calibrations for ultra-high dose array beams. A little bit of housekeeping. We are recording the webinar, so watch your email for a link to the recording once that's completed. Old webinars are also available on our website, or if you want to refer a friend to this, send them to our website. They can get to the webinars as well. If you have questions, please ask them in the Teams Q&A dialog box. I think the chat may also be open for this. Some people are a little more comfortable with that, but the Q&A sometimes works a little bit better for us on this end. If you are not an English speaker, please feel free to use the Teams features, turn on live captions or just the caption languages. Please just don't change the spoken language, because that'll change it for everybody, and then the transcriptions will all look strange. Acknowledgements and disclosures for what I'm about to talk about, things I really do think you should know. Some things we're obligated to tell you, obviously, but you should know the Exradin is a work in progress. It's not yet a commercially released product. If you're interested in collaborating on the final validation and testing of this, please contact me. My email's up there. I think I put my email at the end of the talk as well. Our Exradin A30 does have a patent pending. We're excited to have that filed as well. And the project has been funded by a couple of small business innovation research contracts through the National Institute of Health and the National Cancer Institute. So very grateful for those opportunities. And even more grateful for research collaborators, especially the group at MD Anderson Cancer Center and the one at the University of Wisconsin-Madison. I will point out special thanks to Kevin Liu. Most of the data that you'll see in my presentation was his measurements. So thank you to all of those collaborators. Brief introduction to Flash. I'm guessing that 99.5% of those of you who've joined me on this talk already know what Flash is. So I will be brief, but I do like to have us all on the same page just for the key points that I want to touch on. So ultra high dose rate Flash irradiation was shown to provide equivalent tumor control with decreased normal tissue complications compared with conventional dose rates. And if you have not read Favedon's paper, I do recommend that initial rediscovery, I guess, but recent publication that kicked off all of this interest in Flash over the past 10 or 11 years. It's definitely worth taking a look at. It generated a whole lot of excitement in radiotherapy. And of course, the first thing we start to ask as medical physicists is, can we push the doses a bit higher, or the physicians as well, right? Can we push the doses higher so that we can have improved tumor control without increasing the normal tissue toxicities that patients are already being asked to deal with? And of course, what's the dose rate threshold? There's a whole lot of investigation into the why and the how in order to get the Flash effect. But key on this really is the Flash effect is a normal tissue effect. And so the dose rate in the normal tissue is really what matters most for achieving this Flash effect. And in order to really ensure that that's happening, we need to know the dose from these beams, and we need to know it accurately. So as with the single high dose fraction treatments like stereotactic radiosurgery, SRS, there's not much room for error here. We need to have accurate measurements for these doses. Ionization chambers that are already on the market, as I'm sure all of you know, have significant recombination and saturation effects that show up in these ultra high dose rates. So most people are turning to other dose rate or dose per pulse independent detectors for their beam calibrations, using radiochromic film, using alanine, TLDs, OSLDs, even calorimeters. If you're at the National Standards Labs, they're a little more proficient with that than some of us out in the field, unless you can get the graphite probe from the is it the NRCC. Those sorts of things are a little easier, but it's still a tricky business. Most of these detectors are not necessarily real time. Some of the calorimeters are. Most have to be read out after the fact, and so you give your dose, and later you find out what the significantly later you find out what the doses are. So the ion chambers, really, as medical physicists, those are really our detectors of choice. Still, it's an active measurement system. It gives you a result right there in real time. They're simple to use. I mean, obviously, some correction factors in terms of linking the charge that's measured directly to dose through energy response and all of that. But we're familiar with those, and so it feels a lot simpler than some of the other systems. But most of all, they can be calibrated at the national or at the dosimetry calibration labs so that you can get values that are NIST traceable and really trust that you're measuring dose accurately for these beams. So the challenge for us was, can we create an ion chamber that has minimal saturation and can be calibrated at an ADCL? The general challenge of dosimetry in terms of the function of ion chambers, why the current ones don't work very well, really centers on that high recombination. This is my fancy parallel plate chamber here. Sorry for my expert drawings, in expert drawings. So you have your entrance window, you have your collecting electrode. We like to collect negative ions in general, just the way the calibration labs do. So that's the way I've drawn it here. We have an ionization track through this air cavity, and we create ions. Initial recombination for a really high dense track, that would be these positive and negative ions recombining. When you have a high density of tracks, I didn't make a picture, sorry, you can imagine there's a very large cloud of then those negative and positive ions within the air cavity of the ionization chamber. And because there are so many charges that are generated in such a short time, you get some self-shielding of the ions within the cloud from the electric field of the parallel plate chamber. Or cylindrical chamber, but in this case, parallel plate chamber, that reduces the electric field pull from the electrodes on those ions, which gives them more time to recombine with ions around them from other tracks or from their same track. That's volume recombination. The larger your plate separation with these parallel plate chambers, the bigger the problem is. There's also, in addition to this, some limitations with an electrometer for the readout of this device. So you can have saturation due to a really high instantaneous current, and then you have what are called impulse response dependencies. So an impulse is an input signal that's really a delta function. It's a really sharp spike of signal, short rise time, short fall time, with nothing else around it, or if you have pulses, then those are separated. The electrometer, in my cartoon here, electrometer has some sort of rise time that it's dealing with, and then some sort of decay time. And so this impulse input becomes a more smoothed out curve with your electrometer to a more or less degree, greater or lesser degree, depending upon your electrometer. This can also cause some issues if the input signal, that impulse, is extremely high. It might be truncated a bit by the electrometer, or if those pulses are too close together, then you get an overlap of the rise and fall of the electrometer. Again, based primarily on the magnitude of that input signal. So ideally, you would have this full area under the curve here matching the area under this sharp impulse, so that the data you collect match the data that went in. Those are the challenges, right? In flash irradiations, we've got generally the proton, the electron, and they're working on photons. So the type of particle, we can measure all of those with ion chambers. The type of particle isn't as big an issue. The way the pulses are generated and the pulse repetition frequencies, or PRFs, that's what really plays into your ion chamber response. So for protons, in order to get to flash dose rates, a lot of proton systems increase the pulse repetition frequency. It wasn't a major increase in the pulse height, but you're jamming those pulses all together into a short time frame, which generates what's often referred to as a quasi-continuous beam. Those pulses are so close together that the measurement systems really see that as an almost continuous beam. Some of the protons, especially if you're talking synchrotrons, you've got a fine structure of micropulses within kind of larger macro pulses or dumps from the synchrotron. But in that case, it's the macro pulse, the larger pulse grouping that really affects your ion chamber response. For electrons, most of the systems for flash have extremely high dose per pulse output with just a few pulses or more spread out, lower pulse repetition frequency. And then photons, to my knowledge, that's still in development, but what I see of the systems that they're talking about developing, that's most likely going to be more of a quasi-continuous beam as well, of very closely spaced pulses from photon systems. So really, again, in our ion chamber development, the electrons provide the biggest challenge and the most likely to fail, the hardest issues to deal with in terms of managing the ion chamber response, managing electrometer impulse response as well, so keeping the signal within the measurable range. So to design a new chamber, we had some ideas, both, and expectations that we wanted to meet for a new chamber. There were a few prior publications when we started into this project that proposed ultra-thin parallel plate chambers as a possible solution. So as part of our initial work, we did some 1D MATLAB calculations to confirm that yes, indeed, that looked like a promising way to go, at least within the simple model of the 1D MATLAB simulations. But construction challenges then are the next big hurdle. How do you make parallel smooth surfaces when you're talking sub-millimeter separation of the parallel plate chambers? It has to be accurate plate separation so that every time you manufacture a chamber, it's the same thing. The manufacturing tolerances on this sort of a project are extremely tight. And then we had to choose materials that we trusted, that we knew would not give us high energy responses, energy dependency in the response. So trying to avoid the high Z materials, and also wanting high dose resistance, because we know in flash beams, we're giving a lot of dose very regularly to these chambers. So we want them to be able to last for a while. We need it not to just fall apart and stop measuring accurately at low doses. So design decisions about the chamber: how big are we making it, the chamber volume, if we're talking about a parallel plate chamber where we're kind of fixed at our target of 0.3-ish millimeter separation. We control the chamber volume by changing the electrode diameter. We'd like to have something that gives us a large enough signal that it can be calibrated at one of the accredited dosimetry calibration labs, but small enough that we're not saturating all of the electrometers that are on the market, particularly ours, of course. But it's not saturating the electrometers when we're doing these flash irradiations and flash measurements. And then, of course, you want to put it in water. Can we make it waterproof and still make it something that is just as repeatable to construct and just as robust to dose? So the prototype chamber design, most of the images on the plots I'm going to show you here come from this publication from Liu et al. I have the link for it here as well. Thank you to Kevin for all of the work on this manuscript for measurements and getting this to publication. But the first challenge that we had in creating prototypes was evaluating that plate separation. So we created a prototype chamber based on our A11 parallel plate chamber. And we created replaceable caps for it. So this step function in the image here really was just an attempt to try and make one image that showed we had different caps so that we could have everything from 0.2 of a millimeter up to a 1 millimeter plate separation. They weren't all on the same chamber. this was 5 separate caps, 6 separate caps. I can count 6 separate caps for this one. So we took that chamber and investigated the effect of changing the electric field strength for each of these different plate separations. And if you look at the electric field strength versus the signal normalized here to this 1,000 volts per millimeter electric field strength, once you get above 12 to 1,500 volts per millimeter, we start to move into the charge multiplication region of the ionization chambers. So you're creating a cascade rather than just collecting the ions that are generated by the irradiation itself. So we'd like to stay out of that region if we're going to stay in the calibratable region in terms of linking the signal to the dose. So the next prototypes that we made one with just the fixed cap, not replaceable, at 0.3 millimeters. In going back to this variable gap, the 0.2 was just a little bit too tight in terms of the manufacturing tolerances. 0.3 was kind of the Goldilocks spot where we got really good response but we could construct it well. And once we got higher than that, the behavior of the chamber was not quite where we wanted it to be. So we made that A11 thin parallel plate. That one has the 20 millimeter collecting electrode diameter matching our A11, which gives it a 0.3 millimeters gap. That's a 0.093 cubic centimeters volume. We also made one with a smaller diameter, and that's the one that ultimately got named the A30. It has a 5.4 millimeter collecting electrode diameter, which puts its volume then with that 0.3 millimeter air gap at 0.0075 cubic centimeters. That's almost exactly in line with our A16 microchamber, the smallest microchamber that we make. So we were relatively confident in making this prototype that we would have enough signal in a conventional cobalt beam to enable calibration of this so that we can link back to established calibration NIST-traceable methods with this chamber. So we revisited that electric field strength measurement with the fixed gap chambers, the A11 thin parallel plate and that A30, and validated again that yes, as long as we're staying in this region, we're outside where we get to charge multiplication, but we're nearing that edge, so we can have the strongest electric field that we reasonably can produce within this chamber without stepping over into the charge multiplication. We looked back at that MATLAB model for charge collection efficiency. You can ignore the 500 volts per millimeter. We're looking more at this 1,000 to 1,200. We expect to have nice uniform response as the dose per pulse goes up with this chamber. Now in real life with measurements with the A11 thin parallel plate version and the A30, you can see that we actually had some significant fall off with the broader chamber, the larger chamber, at a lower dose per pulse, where the A30 kept a little bit closer to that simplistic model. This is most likely due more to electrometer impulse response because of the volume of that chamber than necessarily the chamber behavior, but it is an interplay of the two. So going on then with that A30 design with the smaller diameter collecting electrode, we looked at pulse repetition frequency, and at these higher electric field strengths, we get a nice uniform response across a good range of pulse repetition frequency for these high dose per pulse electron beams. We looked at pulse width as well, because the width of the pulse for a certain dose will also impact the response of the chamber. And again, we got good matching with our theoretical models results across a range of pulse widths. Up to the 4 microsecond was the largest we're looking at, and up to 5 gray. We can't get that in the small pulse widths, but up to 5 gray per pulse with these narrow pulse widths, the chamber was performing beautifully, again, at that 1,000 volts per millimeter electric field strength. We performed a dose lifetime evaluation study initially with one of the early prototypes. We really do think that the chamber should last for a while, the release chamber. So with that initial prototype design, we delivered 300 kilogray over a period of months. It was several different irradiations for that one, and checked the leakage value after each of these irradiations, and it was showing no dose degradation whatsoever at that point. Due to a slight misunderstanding, there was an additional 300 kilogray delivered in a single session, and we melted it, which was good to learn. It would have been nice to see how far we could have gone with the more gradual irradiations. But the 300 kilogray, you can see this plastic used to be black, where heat and radiation damaged it here. This is one of the insulating plastics that we use. Interesting to note here, and what was really kind of a neat discovery for us, is that the conducting plastic, the C552 that we use for the central electrode and the guard ring, as well as this buildup cap material, did not show the same radiation dose damage. And so in the later prototypes, we're replacing this particular plastic with one of the ones that that did withstand that single high dose irradiation. So with the final design, we'll repeat dose lifetime testing, but we do very confidently expect that to show that these chambers should last well beyond that 600 kilogray that this one melted at. So the current status of the A30, which is probably the key point that brought most of you to my talk today, is that we are finalizing and validating construction process for the waterproof design, which is what the release chamber will be. That we have KQ calculations underway with collaborators, and those look beautiful at the moment. I'm very happy with those. And we've been working with the University of Wisconsin ADCL to verify that this is a calibratable dosimeter. They're happy with the signal level, they're happy with the stability and the noise of it, that it will be something that they'll be willing to provide calibrations for with standard cobalt dosed water. Of course, we'll validate that dose lifetime study, or repeat the dose lifetime study with the final production design, as well as just overall behavior of once we have this out of the research construction and into standard production methods, making sure that all of the chambers are identical and behaving exactly the way we expect them to, just like the prototypes were. We're working through regulatory clearance as well in anticipation of release of this chamber, but very excited to be able to talk to you about the research that's been done so far. So that's pretty much all I had at the moment. I guess I have not been monitoring the Q&A. I don't see anything right now. If you have questions, please follow up either with me directly. This is my email address, or through the chat and the Q&A here. Thank you so much for your time. I appreciate everyone showing up for the webinar today, and I look forward to talking to you more about the A30. Any testing at pulse repetition frequencies greater than 150? Not with the electron beams that we're looking at so far, but we definitely will be taking this to protons, and actually it's one of the prototypes was used in protons as well. I don't remember the PRF for that, sorry. But yes, we'll definitely be looking at PRF values that are higher than that. Good question. Not seeing any other questions, I guess I will close the session. So thank you. Appreciate your time. And I look forward. Yeah.
One question in there, Shannon.
Oh, did I miss it?
Yeah, it looks like 2 just came through.
The schematic of the chamber one last time. Yep, I can show you that one. And then, is it more reasonable to use ultra high dose rate for calibration than conventional cobalt 60? That is also an excellent question. At some point, there may be protocols defined for using ultra high dose rate calibrations, but right now, the available NIST-traceable calibrations are the standard dosimetry protocols. So the standard in cobalt dose to water calibrations. And so our goal with this is to have a chamber that can span that gap, that can be calibrated in cobalt 60, but can be used accurately without 80% corrections like some of the ion chambers need within the high dose rate radiation. If we can calibrate it in cobalt and calibrate it in the ultra high dose per pulse radiation, then that's a great validation of that ability to span the gap as well. So I look forward to those protocols, but I know that they don't exist yet. Up to what dose is the detector linear with delivered dose? That depends mostly on dose per pulse. So we've looked at dose per pulse in a 4 microsecond pulse up to about 9 gray per pulse. If your pulse is broader than 4 microseconds, then it's easier to measure the dose at a higher dose per pulse. But the narrower that the pulse width is, the more challenging it is for the chamber. So we were looking at still 95% collection efficiency up at that 8 or 9 gray per pulse. All right, I'm finally figuring out how to see the new questions. Can you get a prototype for research at this time? You would need to reach out to me, and we can talk about the process for getting a prototype. We can't provide one to everybody who asks. It's just not feasible from a business perspective, as well as just we have a focus on trying to get this released so that we can get it out to people. But if you have a strong desire to collaborate with us and maybe take a look at some prototypes or a prototype, please reach out to me and we can talk about that. When you investigate the charge collection efficiency, how do you know what's the real amount of charge generated by the beam? In this case, we were comparing the dose response in conventional beam to the dose or the charge that was measured in the ultra high dose per pulse electron beam and monitoring that electron beam. This was a Mobitron system, although we did some on a converted linear accelerator as well. But on the Mobitron system, we were monitoring the dose that was given to the ion chamber using the toroids that were built into the Mobitron. We also validated the dose using radiochromic film right at the place where the ion chamber was measuring the dose. And then can you advise on the performance for continuous pulse-to-pulse measurements? That was that pulse repetition frequency investigation. So if your pulses are closer spaced, but still high dose per pulse, then the behavior of the chamber should be stable up to a point. If you're doing more of the quasi-continuous measurements, beams like proton beams, generally those pulses are not quite so high, and it's a lower dose per pulse, so that the problem is an easier problem to solve. The saturation happens much later than it does with the super high dose per pulse. Hopefully that answers your question. And then, does the detector begin to saturate as more dose is delivered? Can it measure a large single delivery, and if so, up to what value? Eventually, it will saturate. I don't know what that value is, and it will very much depend on the time in which that dose was delivered. So that the transit time, electron transit time for these type of air gaps with this this electric field strength, I think, is on the order of 3 to 5 microseconds. And so that's going to play into the actual saturation value. So I'm sorry I don't have a number for you, but those are some of the things that play into that. And the p-ion range that I had that, and I don't have it on the tip of my tongue. With the prototypes, I think we were looking at p-ion within, oh, I'm not going to quote a number because I'll quote it wrong. I don't have it in front of me. The p-ion values in electron fields are generally a little bit higher than they are in photons as well. So it did have a little bit of p-ion dependency, but it was relatively stable. And so it is something that can be corrected for, or you use these the same way that that you do microchambers, where you don't apply necessarily a p-ion, and you just use them in the configuration that they are for calibration. So the exact recommendation on that will have to come from the research community, I think, more than from us, but we are trying to make it with as a minimal p-ion as much as possible. It is a little difficult in a parallel plate chamber with electron beams just because you generate constant currents. It's part of the nature of the beast. So I'll go back. Whoops, that's not what I wanted. I was going to go back to my question slide just so that you guys have my email address one last time. That's what I was looking for. And then I think I've gotten through all your questions. Thank you once again. Excellent engagement. I do really appreciate the questions. And I look forward to talking to all of you again.